Hello, my name is Athanasios Zovoilis, and my lab is located at the Southern Alberta Genome Science Center at the University of Lethbridge in Canada. Firstly, I would like to share some disclosures with you. And I would like to start with thanking the organizers for the opportunity to present during this year's ISMB an approach of direct nanopore RNA sequencing that my lab has developed, which provides additional tools for people that study non-coding RNAs. For this audience, I think providing many details regarding nanopore sequencing may be redundant. Nanopore sequencing does offer long-read RNA sequencing, but one specific property of nanopore sequencing that has received particular attention during the recent years is the potential, theoretically, to identify RNA modifications on the RNA strand. And there has been actually recently a number of proof of principle papers that show that this is indeed possible, which significantly expands actually the capabilities for studying RNA modifications using this platform. Direct RNA sequencing library construction shares some similarities with poly-A selection Illumina sequencing, in that actually the RNAs that are selected for this process need to have a poly-A tail, and this poly-A tail is then used for ligating an adapter that has a poly-T tail, and subsequently ligating to that adapter the adapter with the motor protein that will enable the direct RNA sequencing. And this poly-A tail here can be seen as this form of plateau in the respective squiggle. This approach, as I mentioned, has already proved to provide the potential to identify RNA modifications in RNAs tested with the standard direct RNA sequencing protocol from Nanopore. However, one question is, can we identify all RNAs in the Nanopore direct RNA-seq data? Because it makes no sense to have the tool to identify RNA modifications if, however, you cannot identify a specific RNA that you may be interested in this data. And this is of particular interest if we take into account some characteristics that many non-coding RNAs share. So we know that in the mammalian genome, 2 to 3% is either mRNAs or ribosomal RNAs, tRNAs, and other structural RNAs. But we also have a large part which is transcribed into other types of non-coding RNAs. And although mRNAs do have a poly-A tail, and many of non-coding RNAs, of long non-coding RNAs, do also have a poly-A tail, there is still a big number of other RNAs like tRNAs, no-RNAs, snRNAs, as well other types of non-coding RNAs that do not have this poly-A tail. Moreover, some of these RNAs are usually of smaller size. They're shorter than their longer counterparts. And this generates a number of challenges because we're not really sure whether these RNAs are identified with the standard direct RNA-seq protocol, although we do actually assume that this may not be possible, because if they don't have a poly-A tail, we don't really expect this adapter to ligate there. And this was a question that for students in my lab have been working during the recent years, Luke, Babita, Yubo, and Liam, and they particularly have worked on the question how we can actually improve the standard direct RNA-seq protocol so that we are able to identify more non-coding RNAs that may be missed through that protocol. And with that, we have developed the non-coding enriched RNA direct sequencing, which for short I'll be referring to as NERD-seq. So how can we do this? In order to understand what improvements we should make to this protocol, we first have to understand which are potential bottlenecks in the standard protocol regarding identification of many non-coding RNAs. And one particular, first of all, bottleneck is size enrichment. And this is because very often standard RNA extraction protocols do miss many short RNAs that may be less than 200 nucleotides, which, for example, means that we may be missing tRNAs. And tRNAs are known to be very highly modified, so there could be interest for many researchers to identify potential modifications on those RNAs. Another limiting factor, as I mentioned before, is the absence of a poly-A tail. And this absence is actually the most significant limiting factor in this protocol because many, for example, SNO RNAs, as nRNAs, or tRNAs lack this poly-A tail, although we have to mention here that when many of them are marked for degradation, they may get one. And here we have actually a potential of the standard protocol generating some biases here, because those tRNAs or those other short RNAs that people may be studying, and the modifications they will be able to study on them, may be actually connected with a specific biological context, like, for example, degradation of these RNAs. So the same tRNA, for example, that is not mapped for degradation may have different modifications. And this information is getting lost with the standard protocol. Of course, there are additional problems, especially highly structured RNAs like SNO RNAs and tRNAs do generate reverse transcription stops. And although the direct RNA-seq protocol does not depend on sequencing cDNA, they do have an optional, which, however, is almost always used, option to generate a cDNA strand to stabilize the molecule so that it can go through the pore. So absence of this secondary strand very often results in truncated fragments going through the pore. And of course, many non-coding RNAs do have these structures that prevent the reverse transcriptase enzyme from achieving this. Of course, another issue sometimes is that some of these non-coding RNAs may be low expressed. And until recently, the main platform that have been used from people doing nanopore sequencing has been the MinION, which has, however, a low yield of 1 to 5 GB. But a few years ago, with introduction of from Nanopore of a more high yield sequencer, the PromethION, that problem is also to some extent mitigated. So these were the main challenges we have to solve here: size enrichment, poly-A tail non-present, high structures preventing passage through the pore, and RT stops that also generate structural constraints and very low expression. And here's how we try to solve them. First of all, we applied a size enrichment approach that separated shorter fragments from longer fragments. And the reason for that is that in the longer RNA, in the longer fraction, we actually get ribosomal RNA. And as you will see, this is important for our next steps. So we enriched for shorter RNAs in the one fraction, and we also used PromethION in order to increase our overall yield. Then, only in the short RNA fraction, we added poly-A tail. And as I mentioned, that was not a problem for we didn't do this in the longer RNA fraction because in that part, we do have the ribosomal RNA that we don't want to sequence. In order to resolve the structures that could stop the RNA from going through the pore or stop the cDNA strand from covering the whole RNA, we use an enzyme that makes cDNA synthesis at 70 degrees. And overall, with that approach, starting from a total RNA, we have been able to generate the short fraction, and you can see here for the short and the long RNA fraction, the respective electropherograms. And then you can see that by adding in short RNA fraction, the poly-A, we practically extended those short RNAs, and by combining them, we get this compound electropherogram for both the short RNA poly-A fraction and the long RNA fraction. It's significant here to mention that this here, the short RNA part, was practically an enrichment set. So we do get still longer RNAs in that short RNA fraction, but in smaller numbers than in the long one. And here's the overview of our approach. You can see here the standard direct RNA-seq approach with the ligation of the poly-T adapter, and of course, disregarding many short non-coding RNAs getting excluded. And then the first run, cDNA synthesis. And here is our modifications that include the separation to the short RNA and the long RNA fraction, subsequently polyadenylating the short RNA fraction, adding the poly-T adapter, so now we include also these RNAs. And an additional step we have is a high temperature first run synthesis. But what we have noticed when we have been optimizing this approach is that because of the high temperature, we did have significant RNA degradation. But this was mitigated by including random primers that have been coated across the RNA molecules and stabilizing them during this treatment here. And here are our results. So you can see with gray the distribution of reads from the standard RNA approach, and with red the distribution from the NERD-seq. And one first finding here, which is something actually that we would expect, is that we have an enrichment of shorter fragments around position 350. And when we compare the standard with the NERD-seq, we see that although the vast majority of transcripts that are identified are coding transcripts, mRNA transcripts, the part of the non-coding identified with NERD-seq is significantly higher. And we can see here for exon versus intronic and intergenic regions and lines and signs and other repeated sequences, how NERD-seq actually provides in the non-coding part a significant enrichment. Of course, NERD-seq can still detect mRNAs, and here is a distribution comparing standard RNA-seq with NERD-seq around the transcriptional start site of known mRNAs, and can still detect non-coding RNAs that we know that have a poly-A tail, like SINE B2 RNAs. So you can see here, but in both cases, and these are relative density profiles, we see that we have less enrichment. So still can detect those classes, but not as much enrichment as in the standard protocol. But where the difference was really significant was when we tested non-poly-A non-coding RNAs, and particularly short non-poly-A non-coding RNA classes. So you can see that SNO RNAs are almost undetectable with the standard protocol, but we do get a significant enrichment with the NERD-seq protocol. The same applies with snRNAs, scRNAs, and the srpRNA. Then we also tested for tRNAs, and as you can see, the standard protocol can still amplify, can still detect, to be more accurate, tRNAs. However, we don't really know whether these tRNAs here that have a poly-A tail by themselves are the same with the tRNAs detected here, because the standard tRNAs, when they are marked for degradation may have this poly-A tail. So again, we do get some, of course, in our case, more enriched, but we don't really know that those here represent the whole story regarding tRNAs. And the same apply for smaller fragments with from ribosomal RNAs that also have been connected with some biological contexts like response to stress, and which are also sometimes targeted for degradation with the poly-A. Now, we have gone through this protocol and identified other class of non-coding RNAs, and unfortunately, I don't have enough time to come into details, but we did find also classes of non-coding RNAs within lines that the standard protocol cannot identify, but the NERD-seq can identify, and that's particularly parts of line L1. So there's still many things that we don't know regarding those RNAs that are identified by our process. And to sum up, using size enrichment, polyadenylation, high temperature cDNA synthesis, and a high yield platform, we have been able to identify mRNAs, siRNAs, many classes of short RNAs, and other non-coding RNAs that the standard approach cannot identify. And with this, I would like to thank the people in my lab, our collaborators, and funding agencies, and I will be happy to take questions.